स्लाइड्स आ रही हैं क्यों यार यहाँ पे क्यों आ रही है?
Good morning, sir. Good
Uh,
morning, everyone.
are the slides in full?
The side slides are visible.
It is in presentation mode.
No, sir. It's not in the presentation. The slides are not visible.
Now. Now, are they in this mode, presentation mode?
Sorry, now it's visible, sir.
Yeah, it is in the presentation mode now. Okay.
Yes, sir.
Okay.
Good morning, everyone. I am your moderator for today's webinar. My name is Rojamani, Director of Virtual Learning in a Pan Pharmacon Student Club, and I will be here to help facilitate the discussion and ensure that everything runs smoothly. I would like to welcome our speaker, Dr. Ishan Pandey, Reader and Head, Department of Microbiology, Babu Banaras Das College of Dental Sciences, Babu Banaras Das University, Lucknow. And I would like to welcome you all to this webinar on Oncobiome, Bane or Boon in the translational research. Before we begin, I would like to go over a few things. First, please make sure that your microphones are muted throughout the webinar to avoid any background noise. Second, if you have any questions or comments, please type them in a chat box and I will do my best to address them during Q&A portion of the webinar. Now, I would like to invite Leah Biju to give a brief introduction of our speaker.
Am I audible, Rajamani?
Yeah, your voice is audible, Leah.
Good morning, all. Welcome to everyone who has joined us for this fantastic and enlightening webinar today. We are excited to talk about the interesting subject of Oncobiome, Boon and Bane in translational research. Myself, Leah Biju, treasurer of Pan Pharmacon Student Club. Before we begin our session, I would like to give a special greeting to our valued resource person, Dr. Ishan Pandey. We are truly honored to have such an eminent personality whose accomplishments in the field of medical microbiology are truly inspiring. Dr. Ishan Pandey is a distinguished scholar dedicated to pursuing knowledge in advancing medical science, having completed his PhD in medical microbiology from the Department of Pathology at Motilal Nehru Medical College. His journey of excellence continued as he was appointed as a visiting research trainee and junior research assistant at King's George Medical University. His contribution to the field were recognized and he subsequently joined the Center for Medical Diagnostics and Research at Motilal Nehru National Institute of Technology. He serves as a scientific reviewer for several prestigious journals, including BMC Genomics, Scientific Reports, Frontier in Bioengineering and Biotechnology, International Journal of Surgery, and many more. Today, we have the privilege to listen Dr. Ishan, a true luminary in his field. I'm confident that his keynote speech will enlighten us all and inspire to reach us greater heights in our own endeavors. So let us welcome Dr. Ishan Pandey with enthusiasm and open hearts, ready to absorb the knowledge and insight he graciously shares with us. Thank you, Dr. Ishan Pandey, for gracing us with your presence, for all you have done to contribute to the field of medical microbiology. Now, I heartfully welcome our resource person, Dr. Ishan Pandey. So let's start the interactive and exciting section and make us use of valuable opportunity. Once again, a heartfelt thank you to all for joining us today. Thank you.
Thank you so much. And shall I start the presentation?
Sir, can you please share the PPT in presentation mode?
Yeah, I've shared that maybe.
It's visible, sir, but not in presentation mode.
Okay, okay, right. Now is it full screen mode?
Yes,
Hello.
sir. Yes,
Yes,
yes.
sir. Yes, sir.
Okay, okay, okay. So shall I start?
Yes, sir.
Okay, okay. Thank you so much all for the warm welcome and saying a lot of kind words about me. And here I go with the seminar or the presentation. It's about the Oncobiome, which is said to be the Boon or a Bane in translation research. Means there has much been about this Oncobiome. It is a relatively new word. There has been a lot of talk about microbiome. We all must have listened about the microbiome. There is about not even about human microbiome. Now there is a lot of talk about animal microbiome. There is plant microbiome, soil microbiome. So what is all about this? But since we are talking about this medical field, so we will be discussing about the interplay between the human microbiome and its role in the carcinogenesis and its implication in the cancer research. So carcinogenesis and the human microbiome. Now the human microbiome is divided into different parts of the body, like the systemic microbiology. But the most of the thing that has been talked about is about the gut microbiome or the stomach, which is the main part of our body, which goes from the mouth to the anus, which is the largest organ of our body. So the stomach is a reservoir of millions and trillions of microbes. So that has been a lot of talk in the recent past decade, and we should start our presentation with that. So, as I was talking about, what is the microbiome or the human microbiome? So, the microbiome represents tenfold higher than other of our cells. Now, of late, there has been a discussion about what is this, what our body is made up of. So, roughly, it has been discussed that our one cell of our human body consists about 10 microbial cells. So we are not just humans or we are not just about cells. It's all about the microbes that are residing in and outside our body, which constitute the microbiome. So it is about 10 folds higher than our cells in the body. So we are much more than the living cells. We are about the microbes that live inside us and outside our body on the skin and the environment. So it is totally that we are about 10 to the power 10 times more microbes that lives in as a microbiome. Now the microbiome includes bacteria, viruses, protozoans, fungi, archaea. These all microscopic organisms constitute to the microbiome. Now the balanced microbial communities can positively contribute to the immune homeostasis. Now there is a balance. Now there is an interplay between those species, the bacteria, the viruses, the protozoans, the fungi, the archaea. They live in harmony. They live in a symbiotic association. They live inside in a harmony in a symbiotic manner. So there may be some beneficial parts of it. There may be some harmful parts of it. So the balance should be always be maintained. Now, when this balance, which is known as the eubiosis, which I'll be discussing on the later slides, I have tried to keep this presentation very smooth and very simple so that all the students and all of them can understand it equally well. And I will try to discuss because this area is very vast and there is a lot of intricacies in the subject, but I will try to discuss what are the basics and what is the loopholes or the means the gaps which needs to be addressed and which needs to be understood in the meantime. So this biases now the what I was telling you that the balanced microbial communities can positively contribute to the immune homeostasis. This balance is known as eubiosis. And when this eubiosis or the balance becomes disturbed, it is known as dysbiosis. Now, dysbiosis is when the good beneficial microbes in the body becomes less and the harmful microbes oversede or supersede its concentration. Then it is called as dysbiosis. Now, these microbes, they have virulence factors, they have cytotoxins, they have genotoxins, they have lots of other virulence factors. These contribute to the chronic inflammation. They may disrupt the epithelial barrier. They may cause different kind of disturbance in the metabolism, because our body is a reservoir or a niche for growth of these microorganisms, because they get all kinds of nutrition, all kinds of things. There are some parts of the body where they get an anaerobic environment. There are some parts of the body where they get an aerobic environment. So all kinds of environment, apart from the outside environment, is present inside the body, along with having the proper nutrition. Now, there have been a lot of talk about the role of oncogenesis, because cancer is a multifactorial disease. There is not a single factor that is contributing to the development of the cancer or in the field of oncology. So, of late, there has been a lot of discussion about the role of microbes in cancer. Reflect diverse physiological or pathological states. So there have been bacteria that have been contributed to the development of cancers. There have been viruses. There are oncogenic viruses. There are oncogenic bacteria. They have been classified. There is an agency at the level of WHO, which is known as International Agency for the Research on Cancer, IARC, which is based in France, Lyon. That classifies the types of carcinogens, which are uh, these biotic carcinogens, which are the abiotic carcinogens, because there are lots of heavy. Now, of late, and apart from you all have seen this COVID-19 pandemic, there is a lot of role of microbes in the progression of different diseases. So the microbes have a lot of role in causing the cancer. Because the oncobiome, now if we are talking about microbes, so we'll give it a better term, because microbiome is a general term, and then we have to classify it as in a human microbiome or animal microbiome. And when we classify furthermore in the human microbiome, we go to oncobiome, which refers to the interplay between the human microbiome and carcinogenesis. So the interaction among the microbiota and the host interactions can cause cancer. As I've already told you, that they may cause inflammation, they may cause immune checkpoint blockages, the, these kind of things they may do. They do. Individual microbial species have been identified as promoting cancer progression. Now, implications for cancer treatment, there as if they can cause cancer, they may also be needed in the means, I mean, the diagnosis of cancer. They may be helpful in immunotherapies. They may be boosting the therapies. They may be the slowing down the therapies as well. So the role of microbes inside the gut, because most of the drugs, as this department you will be, I'm conducting this seminar, is of pharmacology. So you all must be aware of it, that when we take drugs, and the most common route of administration of drug is oral mode. So when we take oral mode drug, that goes from the mouth towards the stomach, and it goes. So there is a lot of the microbes that are residing inside our stomach, in the intestines, that helps in the pharmacokinetics as well as the pharmacodynamics of the drugs. Now these drugs the microbes can influence the efficacy of anti-cancer drugs as well. Now, the identification of microbes of drug resistance in cancer patients has been subjected to numerous microbiome studies. Means there are lots of studies where the drugs which have developed resistance, or the cancer resistance from the drugs, have been developed due to the resistance. And there is a common term, or a term which you all must be familiar about, the antimicrobial resistance, which is quite similar to the drug resistance in oncology. Now this antimicrobial resistance being developed by these microbes has been due to the lot of environmental changes, due to other changes. So the bacteria, or the specifically the microbes, are making a synergy with the environment to adjust themselves and pose resistance. Sometimes they are accumulating drugs inside their system to make a drug resistant system. Now, what is the origin or the cause of oncogenic maintenance of certain cancers? Maybe better explained by the oncogene addiction theories or the genetic alterations. You might have heard about this horizontal transfer of the genes and the genes that are these transposons, these plasmids inside the bacteria, or in the case of viruses, these viruses also transfer their genetic material or integrate inside the cells. So these kind of things are most important at the level of genetic alterations, which causes mutation or changes in the system of the cell to make it oncogenic. Now, how to study these in case of cancer? There is a lot of study about when we talk about cancer. We say that there are two ways. One is an invasive way of diagnosis or treatment, and the other is non-invasive way. So the invasive way is quite costly, and it takes a lot of pain and efforts for the clinicians to do. Now, apart from chemotherapy, or when we go to radiotherapy, or finally when we go to surgery, there are lots of investment and lots of pain involved in this study. So nowadays, for early detection, for time saving, there have been studies regarding metagenomics, metatranscriptomics that can use the non-invasive routes for to detect the biomarkers at a very early stage. Because the most important problem with cancer treatment is the diagnosis. As soon as we diagnose and we get what is the exact reason or is this forming a cancer, should be at a very early stage. If it goes to the second or a third stage, it is hard or almost impossible to treat the cancer. So microbial signatures, what we call, as we call it a biomarker, we can call it a microbial signature as well, where we can metagenomically or by the use of computational methods, by the use of different body fluids that are just secreted without any invasive technique like the saliva, like the urine, like the stool, these kind of things. We can just get in a view of is the progression of cancer in the body. There, of late, there has been two new rules also. When there has been a test in some country in United States, they have given a urine biomarker test, paper-based urine biomarker test to get a view about 90 types of cancer non-invasively. Yes, that's a paper strip test. And secondly, of late you must we all have must listened about this alternatives to animal testing. So the use of in vivo models and animal these kind of cell-based techniques, these metagenomics, metatranscriptomics, these kind of studies have paved our way more human relevant, and this can give us a more non-invasive way or a way. Now the use of artificial intelligence technologies, the use of machine learning technologies have also paved the way for the implications in cancer treatment. Now, as previously I was telling about what is the eubiosis or dysbiosis. Now, eubiosis is the healthy maintenance of the body's microbiome or the human microbiome, and specifically, if we talk about the highly diverse and most abundant microbiome, that is the gut microbiome, which also constitutes to the gut immunology and the immunology of the body. Now, there is the system of this is divided into two parts. That is, one is eubiosis, and the other is dysbiosis. Now, the eubiosis is the maintenance of the good slide. There is represented about the cancers. There are the gastric cancers about the gut, or the breast microbiome, or the tumor microbiome, which we call as the tumor microenvironment. That is a common term which you have just heard about. That is a tumor microenvironment, because the environment residing between two cells, that is the inter means between two cells, and inside the cell. So there is a environment or a micro niche or a micro habitat that is being maintained, and it causes different kind of cancers. Now you can see, I won't go into the lot of detail about this slide, because as I've already told you that I will keep my presentation as simple as that to let understand all. So there is well that in the red it is written about dysbiosis, which causes the uh, means deadly microbes or the harmful microbes to supersede the good microbes. Now, the good microbes may secrete, they have metabolism, they may secrete anti-inflammatory microbial associated molecular patterns. That is the MAMPs, you can see. Now, the SCFAs, which is the short chain fatty acids. You all must have heard about these omega-3 fatty acids and short chain fatty acids. These are antioxidants, and these are the short chain fatty acids which contribute as a good effect in the body, and it causes these and it works as a anti-inflammatory molecule, and it causes a lot of antimicrobial effect as well. There are lots of bacteriocins also which is being secreted by the good bacteria, like we have probiotics. There is a common term about probiotics. Now these probiotics secretes short chain fatty acids and lactic acid bacteria, which is the most common probiotic bacteria found in the curd. So that is why it is being recommended in our traditional system, in our food system, to have curd uh, with our food. I'll be discussing that in the future slides also. Now, these anti-inflammatory MAMPs, and short-chain fatty acids, they uh, cause reduced inflammations. They have microbial byproducts, as the microbes do metabolism. When we eat food, it is a complex in nature. Then that food is broke down into the microbial byproducts. They cause estrogen signaling. They have epigenetic regulation. They have gut barrier homeostasis, metastasis, angiogenesis. These all things are being down-regulated, and few epigenetic regulations are being up-regulated. Means it is uh, means moved up in the way, and also the pro-inflammatory MAMs and the other parts also, they promote the tumor suppressive genes, which is the the dysbiosis occurs. In the dysbiosis, the bacteria which may cause severe effect or the genotoxic effect or the secretion of toxins, they cause lots of uh, pro-inflammatory molecular associated molecular patterns. So microbial associated, as it was anti-inflammatory effect or the positive effects in the eubiosis part. But as soon as it gets dysbiosis, now there are lots of commensal bacteria or the fungus which are residing inside our body. That is, the most common of them is E. coli, or the most common cancer-causing bacteria, or the bacteria known for carcinogenesis, which has been most studied till date, is Helicobacter pylori. So WHO has when I discuss about this in the later slides, first about this oncogenic effect, and when it gets about dysbiosis, I thought earlier, there are the generation of reactive oxygen species, the genotoxins, disruption of the gut barrier, there are endotoxin, exotoxin, there is a formation of biofilm, there is a DNA damage, lots of proliferation, metastasis, all these kind of process that contribute in the development of cancer is upregulated, and they are marked in lots of quantity and in number. Now, what is as I was previously also talking about the approaches or the metagenomic approaches, how we can non-invasively conduct the study on the cancer, because the most important part is that when we enroll any patient or when we enroll any study, the most important thing is that we should do a non-invasive or a study that is most feasible and human relevant. So now the impact has been shifted to the bioinformatics or the computational approach or the non-human or non-animal based approach. So the impact of next generation sequencing, which is nowadays known as next generation sequencing, has come away. So the this next generation sequencing, what it does is it has a sequence or the data of the genetic material of the bacteria or the virus or the part that has been of their concern. So this has revolutionized the study of the microbiomes and the study of the specific species, which are the newer species. Now, if there is an unknown species and we want to study is that bacteria is good or a bad, is it conferring a proper effect, good effect, or a bad effect, can also be assessed by these next generation sequencing technologies because we are having a database and it reduces much more of our time and problem to solve. So to access these bacteria now, you all must have heard about bacterial identification or microbial identification. It takes a lot of time to culture those bacteria. Even there are only 1% of the microorganisms in this universe that can only be cultured. So the 99% of the microbes which cannot be cultured, now the culture-free approach, this metagenomics or the culture-free approach by the use of next-generation sequencing methods is the culture-free approach. So this can take the sample directly from any site and it can give you the exact quantity. Virus means the load of the microbes, what is the quantity of those microbes, and what is the type of those microbes, the similarity of those microbes to the already present microbes in the data, whether it is a new strain of bacteria or a new strain of virus or a new strain of fungus. So these and also how to maintain these data for later use for later study is also very important. So the impact of next generation sequencing in this area of microbiome studies had put a lot of effort to make this study more relevant. Now, as I'm talking about microbiome, then I'm talking about next generation sequencing, I'm talking about eubiosis, dysbiosis. No, those are the contributing factors in the microbiome. As I already told you that microbiome is a composite term. We can talk about plant microbiome also, we can talk about soil microbiome about, and when we are talking about human microbiome, that may be good and bad as well. But when we talk about only the bad part of the microbiome, it contributes to the development of cancer. Now we go to the term of oncobiome. What is oncobiome? Means those biome or those microbial communities that may be responsible to to develop a cancer. Now, oncobiome is the microbiome in cancer patients. Means when we do a study with humans and we take a normal healthy a human, and then we take a patient which is having cancer, then we study about the microbiome composition of that patient with the healthy case. And then we will find, as I already told you, that there will be microbial signatures, there will be a difference as we used to do for the biochemical parameters or the clinical pathological parameters. We will also go it in the way of microbiological patterns also. Now, what is this oncobiome? Now, when we were not only studying the biochemical or the pathological parameters, we will study about the prevalence of the bacteria or the microbial communities that are residing inside the healthy person and inside the cancerous person. So it includes the microorganism in the tumors, in the adjacent tissue, in other parts of the body. What is the exact composition, and then we can compare or correlate uh, whether we can get an idea about the exact diagnosis or the early diagnosis, as I already told you, because for the deaths with cancer are rising and alarming. So the earliest diagnosis or the earliest way of diagnosis is the most important thing in this research of oncology, in the research of oncology. So much is still unknown about the role of microbiome in cancer. As I've already told you, only around two bacteria. One is Helicobacter pylori that has been well known to cause cancer, and the second, which is being in research and has been fixed, that is about Clostridium difficile. That is the second bacteria. And if you go into the viruses, you have human papilloma virus, which is causing cervical cancer. HPV is known to cause cancer. Others may be contributing, they may be cofactor, they may be co-carcinogen, they may be other factors. But the study of oncobiome is still in its early ages, and as discussed, and I will be discussing further also, that it is having a lot of effect on cancer development. So what we will be discussing is this microbiome is having a direct impact on cancer progression, or is it having a cofactor? Is it working as a cofactor, or is it working as a multifactorial effect in the disease progression? So that is the most important thing which is in question, which is of late being discussed, whether the microbes are directly impacting. But it is also in the case of the treatment also, like there are the different kind of live biotherapeutics. Now there are discussion about biotherapeutics. What are the biotherapeutics? Means the live bacteria, the live viruses, like the vaccines, the bacterial vaccines, these are being also administered, the probiotics, the symbiotics, the postbiotics. These kind of terms have also come into the picture, which confer positive effects. So there has been a lot of discussion, there has been a lot of talk about the role of microbes in the development in this field of oncology. Next is, how does the oncobiome affect the cancer? Now, the oncobiome can affect the development of cancer by altering the immune response, as I already told you, because in the case of COVID-19 also, everybody must be aware of the immune response or the cytokine storm. When there is a foreign agent inside the body, like if you have a pathogen, if you have any kind of thing, there is a strict immune response by the body. And when this immune response is being altered, or this immune response is being surpassed, then it can cause cancer. It can also reflect on the response of cancer treatment also. As you all are students of pharmacology, you might be knowing that there are drugs which may be altered because the bioavailability of the drugs may be biotransformed. That is the main important term, that is about the biotransformation. When the drug goes inside the body, these bacteria, these metabolites or the bacteria, they biotransform the drug into an active form or sometimes into inactive form. So that may be a decision that we cannot make, how the drug may be reacting inside the body according to the microorganism. So sometimes those microorganisms may contribute into the positive part, and they may biotransform the drug into much more positive form and may boost the effect of those drugs. But at times it may dysregulate or downregulate, or just even make the drug inactive, and that drug is only passed on to the body, or it may remain inside the body and may cause toxic effects. So there is a study about how this drug and microbe interact. So there is a drug microbe interaction also that is going on inside the body, and lot more research is going on in this area. How this and this is all about the pharmacological dynamics and how these drugs are conferring the effects inside the body. Certain bacteria, as already told, may promote cancer growth while others may inhibit. More research is much needed, and this is in a nascent phase of discussion. So, however, it has the potential to be an important target for cancer treatment. As already told, when there is a dysbiosis, the use of antibiotics, the use of other treatment regimen to suppress those dysbiotic community of the microorganism may be a part. Now, as I'm talking about eubiosis, dysbiosis, microorganism associated molecular patterns, how this now there is a this is a part I've tried to show, that how the different part of the body and what are the microorganisms that are related with carcinogenesis. They you can show that how this cause. So in the intestine, also in the stomach, you can see that bacteroids, fragilis, Helicobacter pylori, and acidobacteria, clostridium, lactobacillus. These all are residing inside the gut. Then there is a part of then there is other parts of the body as well. There are Fusobacterium, Escherichia coli, Shigella, Klebsiella, Clostridium, Streptococcus. All these are associated in the parts and are labeled by these arrows. Now you can see the microorganism associated molecular pattern on the left side, where you have cytotoxic necrotizing factors, toxins. There are reactive oxygen and nitrogen species. They may cause DNA damage, oxidative stress, cell proliferation, angiogenesis, inhibition of apoptosis, and leading to chronic inflammation and causing carcinogenesis. Now the next part is when we come to the right side of it, we also have the same way when in the stomach we talk about the H. pylori, as I already told you. This H. pylori is a potent carcinogen which has been declared by WHO as a type 1 carcinogen in 1994. The so this secretes some virulence factor like the CAGE, VACE, UDAs, NAPA2, and hypochlorhydria, because we are having HCl inside our stomach. So when the quantity or the concentration of this HCl becomes low, the antimicrobial property of this acid becomes less. So that part, the bacteria grows much more and it causes chronic inflammation. And before chronic inflammation, now most of the times H. pylori has been linked with gastric ulcers. So if there is an acute infection or an idiopathic infection, it may cause you the ulcer. Peptic ulcer disease is the most common disease caused by Helicobacter. Now, of late, there has been a discussion about Epstein-Barr virus also, which is also living inside the gut. So there is a relation between, or a type of co-infection occurs inside of, in the case of cancers. So there may be EBV, Epstein-Barr virus, and Helicobacter pylori co-infection leading to the development of this cancer. Now, when we're talking about lung, we go to the right part of this, that Corynebacterium, Staphylococcus, Neisseria, Mycobacterium, Klebsiella, these may also, and apart from that, as I already told you, that what all have been known about oncogenesis or cancer till date is that it is a multifactorial disease. You cannot say that a single factor is responsible for causation of cancer. So there is a tobacco smoke, there may be comorbidities like asthma, COPD, cystic fibrosis, and these all contribute into the formation of chronic inflammation. And again, that chronic inflammation, when this disease persists for longer and longer periods and inflammation is up to a chronic level, it causes carcinogenesis. So again, I would like to summarize the main motto of this presentation is that we are trying to introduce what is the role of these microbes in cancer progression, in cancer prognosis, in cancer diagnosis, or in cancer treatment, or in cancer prevention. So these are the most important points that we need to keep in mind while going through this presentation. And now what has been done till date and what needs to be further done from this part now will be discussed in the further coming slides. So next I move on to the oncobiome studies and challenges. What are the studies? What are the challenges? What are being prevailing in the studies? Are there the study of oncobiome presents a lot of challenges that I already told you, because there are lots of more bacteria or more viruses. Now there is a microbiome, means fungus are also being discussed. Now there is a talk about virome, now parasitome, lots and lots of different kind of organs, the axis, lots and lots of things are being discussed inside the this challenging study of oncobiome, how they are contributing to the complex development of this microenvironment, how they are impacting the genetic upregulations, how they are how they are impacting the anti-cancer treatment regimens. So this also has led to the development of in the area of precision medicine and a personalized therapy. Now, each and every individual is having a different genetic composition. Like it is having a different genetic composition, they are also having a different microbial composition as well. So now microbiome is being considered as a separate identity. It is being considered as a separate organ itself also. The gut microbiome, to be honest, is considered as a second brain, or or the new organ to be discovered, and it is the area which needs much and much more research to understand how these genetic alterations, or how we can study these changes contributing to different kind of diseases. Now the metabolism of these microorganisms may contribute into the diabetes, they may cause neurodegenerative diseases, they may cause lung diseases, they may cause prostate cancer, vaginal cancers. Lots and lots of studies are challenging which needs to be done, and for that we constantly need to work out. Now it is, as I already told you, that only 1% of the microbes are being cultured. So to it, because if we are conducting a live study with humans, we need a lot of ethical considerations, we need a lot of ethical permissions. The patient must die at times. So to enroll patients to get the samples for analysis is a tedious task. So what the next part is that we also need to collaborate for data sharing, for crucial, as I told you, that we need some ethical permissions. So there will be a team, and of late the government of India is being saying about, or being promoting the One Health strategy. Means there has been a team of veterinarians, there have been team of zoologists, there has been team of plant specialists, there have been clinicians, there have been basic scientists, there has been data scientists. All they should integrate to form a One Health consortium that that gives a detail about their expertise and understanding about how this data is needed and how the research should go on. Now, more studies and more studies need to be validated. Now, the most important thing in science comes about the validation studies. Now, getting a study in a mouse model or getting the study inside some patients will not give you a proper clear picture. At times they may be positive, they may be negative, but to get to a conclusion or get to a strict finding, or to make it a protocol, we need a large and robust studies for multi-institutional studies, multi-state study, or a multi-country, even a multi-institutional country level studies where you have uh, collaboration with, like I would share my experience also. I have worked on a project related to human papilloma virus in the etiology of head and neck cancers in India and Europe. So the I was having 16 countries from Europe as well. So those Europe countries were also contributing their data on how the cancer of HPV in the head and neck cancer is prevailing in their parts. Then from India also we have centers from South, CMC Vellore was there, Rajiv Gandhi Center for Biotechnology was there, Delhi State Cancer was there, from Bihar Mahavir Cancer Sansthan was there. So we used to integrate all the data from all parts of the world where there are different climatic conditions, different diet conditions, different means kind of genetic alterations, everything. Then after the study of those robust sample studies from all over the globe, we used to come to an inference that what is the pattern associated with the disease progression, where it is progressing, where it is regressing, where it is less, where it is more. Like it is a very common thing I'm sharing, that most of the times we now talk about viral diseases, we talk about bacterial diseases, but some way or the other we don't talk about parasitic diseases nowadays. There is no talk about entamoeba, there is no talk about GRDR, which used to be a simple waterborne pathogen. Because now we are living a very hygienic life. All of our houses are having these aquaguard system, these RO system. So these have declined the prevalence of these the parasites which were born inside the water of our system. So these kind of organisms have gone down, but the emergence and re-emergence of advanced pathogens like the viruses or the trans species jump from animal to human, from plant to human. These you might have heard lots and lots about viruses these for the past 5 years or so. Like there is lumpy virus, there is monkeypox virus, all are trans-species jumping. Now, the complex nature of this microbiome makes the analysis interpretation more challenging. As I'm already telling you that this microbiome, the interplay between the bacteria, virus, fungus, they all are also contributing. They are also competing in in nature. They are also forming a lot of complex relationship between them, which we call the, and also at last they are having host pathogen interaction as well. Now the host genetic factors and the pathogen, they are also making it more complex to study. So these are the challenges in the study of microbiome. Now potential applications of oncobiome in research. So the oncobiome research may lead to, as I already told you, that may lead to new diagnostic tools for cancer, early detection of cancers, biomarkers, microbial signatures. It may also lead to new therapeutic targets for cancer treatment. Now, when we know that this is the main target, or this is the main microbe that is causing the cancer, we can target it specifically with drugs, with biotherapeutics, and we can have positive effect. However, much more research is needed, and as I told you, that this needs to be validated for further implication. Oncobiome research may also have implications for other diseases beyond cancer. I already told you that it is being implicated in diabetes, it is being implicated in neurodegenerative disorders, it is being implicated in psychiatric disorders, other disorders, and other. It is being related with endocrine disruptions. Maybe it is a thyroid, or it is a diabetes, these kind of things. Now, personalized treatments, already, as I'm talking about precision medicine and personalized therapy, which is being a lot of talk. So individual based, that each person is having a different microbiome, like we are having different DNA, we are having different genetic composition. The microbiome composition of each individual is also different. So based on that analysis, we can devise a system for now, oncobiome and immunotherapy. Now, the most important thing about oncobiome, or the microbiome, or the gut microbiome, which resides inside, now the gut has a system of devising the gut immunology, or the immune system is being derived from the gut itself. Now, oncobiome may affect the response of immunotherapy. There are immune checkpoint inhibitors. There are lots of macrophages. There are the polarization of the macrophages, where they are moving, how the T cells are regulating, how the B cells are being regulated. These are also affected by this microbiome, because certain bacteria have been found to enhance the effectiveness of immunotherapy. I already told you that there are parts that if we give probiotics, antibodies, which is being administered from the outside. There are lots of anti-cancer drugs in immunotherapy, like the perelozumab, trastuzumab. These kind of drugs are being used, the monoclonal antibodies. These bacteria may positively impact them. However, this is also much not validated, that they may be boosting in some kind of patients, in some kind of patients they may not. So the studying the oncobiome for personalized therapy and for personalized regimen is much of the need. So this which is being studied and discussed about the immunotherapy. Now there are targeted therapies as well. Now the devising of these anti-cancer drugs, the nano-based drugs, so gene therapy. These are all the things which needs to be addressed as well. But in case of immunotherapies in the gut derives the immune system, and this resides in the gut and different parts of the body. They may cause positive effects as well as negative effects also. Now the oncobiome in relation with the different axis of the body. Now it has been discussed, and in common terms we have been discussing about lots of things, that there is a relation between one part of the body, and when you go to a clinician, they may give you a drug that he may say that you are not having a good digestive system, and you are having some problem in the digestion, and that may be linked with other part of the body, like there are gut brain axis. And as I already told you, that the gut microbiome is known as the second brain of the body. And at most of the times we used to use a common line for it, that our mind is not working because we are having we are hungry, or we are not having food inside our stomach, so our mind is not working. So this is also a common term in our society as well, and that is being scientifically validated, that there is a connection between the gut and the brain. Now, there is a connection between gut and lung also. There is a connection between gut and skin also. There is a connection between gut and mouth also. And of late, there is a connection between gut and the vaginal axis also. So these are all the axis where the gut and the gut microbiome plays a role, where there is a transfer of the microbes from these part of the body, and the eubiosis or the dysbiosis may cause different kind of phenotypic expression in the skin, lung, brain, vagina, or the oral axis. You may have periodontal diseases, you may have gum diseases, toothaches, lot of bad breath. These are also implicated by the perturbations in the gastric biome. Now, as I already told you, there are about kind of axes which are being much about being discussion, which are being much about study. But the gut-brain axis, as I told you, that it is the second brain, or it is the new organ system of the body, has been discussed much and much more. Because as I told you just now, that when we are hungry, we say that it is all about. It is all that our brains are not working because we are feeling hungry. Likewise, we also say that we are working in Hindi. If you know about Hindi, they say that we are working for our for our food. हम पेट के लिए काम कर रहे हैं, इस पेट के लिए, इस पेट को पालने के लिए, इस पेट को भरने के लिए we are doing work. So that is also being linked with gut and the brain axis. So this gut is playing a part as well in the development of psychological patterns, in depression, in different kind of studies in Parkinson's disease and others. So this have been a lot and lot more about discussion about the brain and the cognitive behavior that are being shown by this oncogene or this microbiome. Now, at times we feel that we are not, we are in depression. We are so in depression. Sometimes people have an increased appetite, they eat much more, they become fatty in depression, and in sometimes in depression they feel totally full, they get devoid of the food, they don't like to eat, they get thin and thinner. So these all the cravings, these all the things are also linked with the microbiome. So this is a very upcoming area and a challenging area. Not upcoming, it has been a lot of talk, and a lot of people are working in the area of neurodegenerative and the microbiome. So this gut and nervous system is a lot of talk in in these days. Now this can also impact the development of cancer, as our psyche or the brain is a central part of our body, and it can put a lot of effect on our system. If at times the drugs also which is being working inside the body work on the basis of the cognition of our system, when we feel that we are getting cured, the efficacy of the drugs becomes much more higher. And if we feel low, that the drugs are not being working and we are not able to cope up with the situation, we may not get cured. So the role of brain, or the role of cognition, role of thought process, the depression, the motivation, the positives, these are also a part in in this development of cancer as well. Now, I was telling you about how this microbiome can help out in different kind of therapy, in drug discovery, in biomarker, in precision medicine, in microbiome therapy. Now, microbiome therapy, I'll be discussing in the next slide. In immunotherapy as well, I was talking about. Now, if you talk about the pharmacokinetics parameters, there is the admit properties, that is the absorption, distribution, toxicological studies, elimination, these kind of studies are being done. So at times these immunotherapeutics, these are being boosted by the microbiome. microbiome derived compounds or the microbiome targeted drugs are also needed for the area of drug discovery. There are biomarkers, microbial signatures, these are also there, computational methods are there, and microbiome therapy. There is a term FMT or the VMT, that is fecal microbiota transplantation or the vaginal microbiome transplantation. Now, you all must have heard about transplant. There is a organ transplant, there is a liver transplant, or a lung transplant, or a kidney transplant. These must you all must have been familiar. Now, the most advanced version of it is the microfecal microbiome transplant. The healthy microbiome, which is being not developed in one person, may be taken from the person from a healthy person's stool, and it is being developed as a medication in a capsule and being transplanted into the diseased person. So this is the newer era where we are using fecal microbiome transplantation as well. Now the microbes are also being transplanted inside the body for better therapy. Now, microbiome interventional strategies, as in the previous slide also I was telling you. Now, these strategies include first and foremost about the diet. Now, the balanced diet, because each and every part in the country as well as abroad is discussing about the diet system. We all have different diet systems. So the balance of those carbohydrates, proteins, how we are taking the fiber content. There is a discussion about the FODMAP diet or the Mediterranean diet, how this diet, and when we come to North India or we come to India, not about North India, when we come to India, we use a lot these lot of spices, herbs in our food. So they may all have polyphenols, they may confer positive antimicrobial effects. So the diet plays a very important role in the distribution of cancer. The vegetarian diet or the non-vegetarian diet, a vegan diet, lots of kinds of diet is also responsible for the progression of cancer. Now, when we talk about diet, when we go to the left side of the slide, there are few terms written, that is prebiotics, probiotics, symbiotics, and postbiotics. Now, that are the live biotherapeutics, which I was talking about, the positive effects. Now, the prebiotic and the probiotic, these two terms go side by side. Probiotics are the, according to me, the most important
Dear participant, sorry for inconvenience. Due to internet issue, we have lost the connection with speaker. We will back soon.
Yeah. Is it coming?
Ishan sir, try sharing the screen.
Yeah. Am I visible?
Yes, sir.
Slides are visible. Hello.
Yes, sir.
The slides are visible.
No, sir.
Okay. Wait. Is it visible now?
Visible, sir.
Yeah. Full mode.
Yes, sir. Presentation mode,
Okay.
it's
Okay.
visible.
Yeah. So should I continue?
Yes, sir. Continue, sir.
Okay. So once again, what I was telling about is about microbiome intervention strategies, about the prebiotics, probiotics, symbiotics, postbiotics. Now, the diet has a lot of influence in our system. We are having a Mediterranean diet or a FODMAP diet. Lots of diet are different from, not even in India. We have cultural differences in our diet, in our food. We are having spices in our food. We are having herbs in our food that may be having polyphenols. They may be conferring different kind of effects like the anti-cancer effect. We are having curcumin in our food that has been well known to have anti-cancerous effects. We have lots of antioxidants in our food. Also, the use of biotherapeutics or the microbes which are conferring positive effects like the lactic acid bacilli or the curd, or there are fermented foods also are there. So now when we talk about prebiotics and probiotics, now the probiotics are the live bacteria that may confer positive effects, and the prebiotics is the food of probiotics. Now, let me give you an example of the common system. When you get diarrhea or you get infection inside the gut, the doctor prescribes you an antibiotic. Now, the antibiotic doesn't know whether to kill the good bacteria or the bad bacteria. It destroys all the bacteria or makes the bacteria very less. Now, to make the good bacteria grow, we need proper time and faster recovery. For that, we give probiotics to increase the number of good bacteria, and also give them prebiotics or the food on which the probiotics thrive and increase their number. So simultaneously, we are increasing the number or the colony counts of those bacteria by two methods. One is by giving them prebiotics to grow, and the second is directly providing the probiotic bacteria inside the body. Now, these probiotic and prebiotic combined are known as symbiotics. Or now the another term which is being used is about the postbiotics. Now, what are postbiotics? Postbiotics are the metabolites, like I'm talking about, that probiotics will use the prebiotic to ferment and secrete short chain fatty acids. So this process may also take time. So the most important part in research nowadays or of late has been the saving of time and money. Most important, why we need to continuously do research and to develop newer strategies, newer techniques, just to save our time and to save the cost of the study, or the cost of the treatment. There are lots of treatments available in the society, but due to the heavy cost and due to the time consuming procedure or the pain associated with the procedure, we are not able to use that process. So to save time and to save money, we need much more integrated approaches like the symbiotics, and then at the later stage it comes to the postbiotics, where the metabolites of these microorganisms are secreted, which are directly conferred inside the body. Now you have the metabolites or the short chain fatty acids, omega-3 acids. There are lots of antioxidants. There are lots of molecules, short chain molecules that have been directly conferred inside the body in spite of these probiotics or prebiotics. Now, as I already told you, there is a part of in the lower right part of it. It is the fecal microbiota transplantation. The fecal microbiota transplantation is about the good bacteria that resides inside a healthy person, and that is being collected or sequenced from the stool of those healthy individuals, and it is being transplanted inside a diseased individual. So that is the next level of transplantation going around across the globe. And then finally comes about the engineering the bacteria, the use of genetic engineering technologies, how we can engineer the bacteria. And this is being known by the term biomimicking or means we are trying to mimic when we give a drug inside the body. That body thinks drugs to be a foreign particle. The bacteria try to suppress it or inactivate it. Our immune system try to evade it. But when we engineer the good bacteria or the commensal bacteria that are living and residing inside our body in harmony, in solace, that bacteria can be engineered to deliver cargo or to deliver the drugs at the site of infection or the site of at the target. Now, to engineering those bacteria which have accumulated the drugs and going to the site, the body will not feel that it is a foreign particle or a foreign bacteria. So that is the next level of engineering of the bacteria and delivering of the therapeutics by the use of good bacteria. Like we are having lactobacillus. Lactobacillus is inside the curd that can be used as a part having drug inside the lactobacillus, and that lactobacillus can go to the target and deliver the drug of choice. So apart from conferring positive effects of the probiotic, it can also deliver genes, it can also deliver anti-cancer drugs, it can also deliver different kinds of drugs. So that has taken the use of genetic engineering or the engineering technologies, artificial intelligence technologies, robotics, these bio-inspired material design. These kind of things has revolutionized our research and the intervention strategies in case of microbiome. Now, cancer therapeutic approach. Once more, I would like to show this that on the above it is written about the diet, probiotics, prebiotic, postbiotics, antibiotics, FMT, VMT, fecal microbiota transplant, FMT, vaginal microbiota transplant, VMT. Then engineering the bacteria. There are different kinds of organisms linked with different kind of the cancers in the body, like there is bladder cancer, cervical cancer, ovarian cancer, endometrial cancer, prostate, colorectal, pancreatic, gastric. Lot and lot more have been discussed, and there are lots of interconnection between them. How the metabolites, how they cause, they confer. There are metabolites of the bacteria which may be conferring good effects also, and there may be genotoxins or the cytotoxins as well. Then comes about the future directions in the Oncobiome research. What are the future aspects of Oncobiome? Now, advancements in the, as I already told you, advancements in the technology will allow for more precise analysis of the Oncobiome. Second is international collaboration and data sharing. As I told you about my study for the HPV, that I have international collaboration with France, Germany, Italy, then in India also east west. That will give a lot of advancement in this study. The potential applications and implications of oncobiome research is vast and exciting. Personalized medicine in the basis of oncobiome is niches, and it and the individual's oncobiome may become a reality in the near future as well. You may be seeing a lots of things that will be done. More studies are needed to validate the findings and uncover new insights into the oncobiome research. Now, gut oncobiome signatures. As I was telling about you, the microbiome signature, there is a project of WHO or the Horizon 2020 of the EU, European Union, that has got oncomicrobiome signatures associated with cancer incidence, prognosis, and prediction of the treatment response. So this, as you can see, the third point, this was funded by the EU Oncobiome. It has 3,000 patients across 10 countries, what I was telling you about. It also investigated the mechanistic interplay about the metabolism, immunity, and this project was a comprehensive study of about 1,500 million euros. And this project played a part in the fundamental knowledge of the novel diagnostic, prognostic test for cancer, as well as tailor-made pre and probiotics. So this will give an accumulating evidence about the role of gut microbiome or the microbiome in health and disease, including cancer. So this will pave into the new way of the study of the first point. You can see that which will pave the way into the study of microbiomics, the omics approaches into the study of microbiome. Now, translating the microbiome, what's the target? Now, as it is a very important, these three, 4 points are very, very important to sum up the story, which we are getting closer to the summing it up. That the most successful translation of the microbiome science in clinical medicine has been the infection of H. pylori. So H. pylori, there have been tests, non-invasive tests like the rapid ultraurease test and the urea breath test, because it secretes a urease enzyme. So its discovery is easy because I have also done my PhD on H. pylori, so I know much more about H. pylori. So there are many non-invasive, and as I already told you, the WHO in 1994 classified as a type 1 carcinogen, but in India it is classified as a co-carcinogen because our diet pattern is different. But in the Western countries, in the US, in Japan, in Korea, they have lots of Helicobacter pylori related cancer because their diet is a smoked diet. They have much more this microwave ovens, the baked diet. This kind of diet causes a lot of this cancer. Then there is a overgrowth of Clostridium difficile also. Not until 2022, the first microbiome therapeutic product, second point, product approved by the US FDA is a refinement of the old drug of the Clostridium difficile, which is also a pathogen. And this is also establishing that there is still much more needed to about the study of this microbiome. So only two, Helicobacter pylori and Clostridium difficile, is the well-known pathogen or the bacteria that may be causing. Now we are talking about stool sample, we are talking about fecal microbiota transplant, we are talking about prebiotic, non-invasive methods of discussion. Then there is also profiling of urine bacteria, urine DNA. I already told you that there should be non-invasive methods. There may be the excreta of the body that may be useful in deciphering the role of these bacteria. So the profiling of urine bacterial DNA to identify an oncobiome in a mouse model has been linked with bladder cancer. Now we are moving up to the next level. In the case of oral microbiome, we are using saliva to decipher the role of this microbiome, or we can predict is the person is having a cancer or a biomarker of cancer through the saliva, and that study is known as salivomics. So saliva, stool, urine, these kind of things, excreta, can be used very well to predict or the prognosis. We can very early tell that is this person is having a likelihood to develop a cancer or any particular kind of disease. So apart from stool is well known, but after that urine is being known, and now we are moving up to the level of saliva as well. Now there is Food and Drug Administration, FDA, there is EDQM and LBP. FDA and EDQM, they have proposed a term about live biotherapeutic product. As I told about you, the postbiotics or the probiotics or these kind of other functional foods, like there are different kind of functional foods are also there, like the fermented foods. There is mushrooms as biotherapeutic foods. These are biotherapeutic products which are being used. Now the next is important thing, the last point, which is about the microbiome clinician and the microbiome clinic, because there is a lot of disparity between the clinical studies or the clinicians they are studying, because they are the ones who are dealing with the patients daily and on a regular basis inside the OPD, in the IPD, inside the they are doing those endoscopic procedures. So they are seeing, so the those are the translational figures. which need to put the strategies of the basic scientists into the translational level. And we should develop a microbiome clinic as well. Like I am telling you from the beginning itself that each and everything are being discussed or being treated up to the level of different biomarkers, up to the level of different profiling, genetic level. But now the microbiome or the gut microbiome or the oncobiome is of the huge concern. We are at every time we are checking the composition of the microbiome inside the body. So based on that, there should be a microbiome clinic which should study the microbiome, and on that basis also we should give a result, or we should give a conclusion on the basis of therapy or the basis of diagnosis as well, because the clinico-pathological correlation is very much needed. At times there is a misinterpretation of the disease, and that is the most crucial point where we lose someone or we gain someone. So the most important part is the diagnosis. Sometimes there is a mishap and we don't diagnose what the actual problem is. So the microbiome clinic or the microbiome clinician should be needed at a part to translate this microbiome research into reality. Now, finally, the conclusion is all about that the oncobiome is a promising area of research with a potential impact on cancer diagnosis and treatment. Much is still unknown about its role in cancer, and much more research is needed to fully understand its mechanism. The human microbiome plays a significant role in carcinogenesis. The potential applications and implications of oncobiome research are vast and exciting. Understanding the microbiome in cancer patients has important implications for cancer treatment. Collaboration and data sharing will be crucial in advancing research in this area. The future of oncobiome research is bright and brightest. So these are my most important references for you all. I may share this slide also. You may check these kind of these references for your thing. And thank you for your time and attention. I hope it was a worth presentation.
Thank you so much, sir, for such an enlightening and informative session. Now the participant can unmute if you have any queries or drop your questions in a chat box.
Very good afternoon, Dr. Ishan Pandit.
Good afternoon, sir.
Am I audible?
Yeah, you're audible, sir.
What do you think that what is the most common cause nowadays? Cancer is becoming so much prominent.
Yeah,
Even
actually,
in the rural area also. The
mhm.
number of patients are increasing day by day.
Yeah. Actually, the problem is that uh, in the rural area, as I told you about the climate, the diet. Now, this has been not only linked with medical sciences, it has gone up to the level of agriculture itself. Now the crops, the food that is being grown, the use of pesticides, the use of herbicides, there are lots of toxicants or the cancer, these elements, heavy metals are growing inside this food and in the crops. So these are also taking a lot of part. The water bodies, they are also having heavy metals. So lots of pollution, microplastics, lots of things, because cancer is a multifactorial disease, and the complexity with cancer is only this thing that we are not able to decipher the exact reason. But nowadays microbiome is playing a larger role. That is why I try to highlight this part. But in, as you said, in rural areas also, now the cleanliness or the hygiene level in rural cities are also not up to the mark. From cities it is going into the rural level. The pollution is much more there, the advancement in technologies is also there, but the use of these crops or the use of these food stuff is a major issue. So agriculture, precision agriculture, and the growth of these nano-based formulations for agriculture is causing a lot of toxicology and havoc in the development of cancer. Because you are exactly right that there are many patients who have never taken smoke or any kind of drugs or any kind of this what you say liver
Yes.
oh sorry
Uh,
alcohol and all, but they are still developing cancer.
yes,
So the
this is
food,
happening. One more
yes.
thing, Dr.
Mm,
Ishan,
mhm.
that lifestyle is also getting changed.
Yeah, that is what
Mm,
I'm saying, that the lifestyle changes is a very much a prominent factor. Because of this development, the lifestyle has changed, and use of electronic devices in the rural setup as well, the circadian rhythm, the clock, their sleeping patterns, much more things have been changed. That is why I told that it is being linked with depression also, it is being linked with motivation also. So lots of problem are being occurring nowadays with these kind of things.
So, what's your suggestion regarding if you have told that a lot of plastics and all things are there in the environment,
Look,
pesticides are there in the
sir,
clothes.
my suggestion is only we can control up to individual level. It is not that one person will go and do. It is the need of the hour. Each and everyone should understand our responsibility. Look, it's a need. We need laptop, we need electronic gadgets, but that should be limited. So the overuse of anything is harmful, or the
Yes,
underuse.
yes,
The
yes.
balance, as I already told you, the balance should be maintained. We cannot say that stop anything. We cannot say that use anything. The balance should be maintained, because each and everything goes by a balance. Even the system or this earth is balanced. So everything is having a balance. When we
Mhm.
break this balance, we get some disturbance.
Yeah. Thank you so much. Best wishes, doctor.
Good afternoon, sir.
Good afternoon.
So this is Anjali from Ramaya, family student.
Yeah,
So I just
yeah,
wanted to know that during cancer, basically doctors prefer not to have sugar. Why is that
yeah,
so?
yeah. Yeah, exactly. Because they don't say about sugar. They say there is a notion that you stop sugar, but that is not about sugar. That is about artificial sugar. You may use jaggery. You may use natural sugar. They say to have fruits. They have natural sugar, but the artificial sugars is having a lot of problems. So artificial sugar and the metabolism of cancer, or the cells those are having this development of cancer, they use these sugar very well to divide and to cause oncogenesis. So that is why this sugar is being restricted, and because also in cancer there is a lot of link with type 2 diabetes, with a lot of fat being developed. That is why sugar is being prohibited. So not all sugar, natural sugar is preferred, but artificial sugar is prohibited.
Thank you, sir.
Sir,
Hello,
Hello.
am I audible?
Yeah.
Uh, Ishan sir, good afternoon. This is Dr. Praveen Dikshit from Kite School of Pharmacy, Ghaziabad.
Yeah. Yeah.
Uh,
Yeah.
sir, first of all, I would like to say thanks to you for delivering such a wonderful topic, Oncobiome.
Yeah.
Sir,
Thank
I have a question.
you. Yeah.
Sir, in our lab, we have a versatile instrument for anti-tubercular studies. Sir, if we
Yeah.
want
Yeah.
to set lab for this study, what is the
Mhm.
preliminary requirement for us?
So what once more, which instrument?
Versatrack we have, sir, in our lab. Versatrack for anti-tubercular studies. Hello, am I audible, sir?
Yeah. I'm not able to listen that instrument.
Our lab instrument
Backtech,
for Versatrack. Versatrack.
Versa track.
Yeah.
Okay.
So, what
So
is the other requirement to set up the lab for working on this bacteria and viruses for this oncobiome research, sir?
Look, it is not limited to any particular instrument. Okay, you can have backtech, you can have whitetech, you can have different identification system also for this bacteria. But for the, as I told you, that there are lots of system. First, you have to identify the individual. You have to go up to the level of the individual, which is having. For that, you need to collaborate with the clinician. That may be initially telling you that this person is having discomfort, or the endoscopic studies will give you the larger picture or the study. Then only you can get to your lab to study about this further study. So you can have backtech, you can have vitech, you can first, and secondly there may be pathogens which may be up to the level of class 2 biosafety. So you need to have a better microbiological facility. Then PCR must be a basic requirement for DNA based studies, and then computational based approach, as I already told, that next generation sequencing, there is fluorescence in situ hybridization, there is western blot. So up to the level of genomic, proteomic, you need all kind of studies. But up to the level of, if you ask up to the level of microbiological level, and if you are talking about tuberculosis studies, you need to basically have a bactech or a vitech to identify the bacteria or the funguses. And other for culturing system, you need different kind of media and setup. So first of all, foremost, you need to have a basic setup. And as I told you, that you need to bridge the gap between the clinical practice and between the basic research system. So you always need a clinical setup with a doctor, with a clinician who will give you the advice on the clinical aspect as well. Then only you can identify the exact causal, because at times there may be association. Let me tell you one incident that we say that Helicobacter pylori is causing gastric cancer, but
Yeah, yeah.
it may not be the reason. Sometimes it may be associated with peptic ulcer disease, and chronically
Mm.
it is associated. Then we
Mhm,
come confer a conclusion that at times it may also conferring a protective effect as well. It may
yeah,
not
yeah.
be leading to form a cancer. So
Mhm.
not every time that Helicobacter pylori or any pathogen is linked with a particular tumor or a particular system is causing a disease. Sometimes it may be protecting it from a disease. Many a times we are having cancer studies that may not having bacteria or the microbes of the concern. So the diagnosis or the prognosis is very complex. For that, you need a team that can decipher the role. So that will be a way, and having the basic molecular setup is needed, and basic facilities for this thing that I told you about the protein study, about if you are going for a clinic pathological study, you may have some histopathological study for the HE staining and all, and then you may be having some immunohistochemistry also. These are not quite costly studies. But for having a larger setup, you need that BSL2 levels facility as well, and backtech, whitetech for fungal and bacterial identification of those pathogens.
Okay. Thank you so much, sir. Sir, I have one more question.
Yeah,
Sir,
yeah.
is there like for informed students, send them to CDRI for their studies of like microbiological studies or some other
Yeah, yeah,
studies
yeah,
on.
yeah. Yeah, there
But
are
is there
lots
any,
of study. There have been study on antimicrobial pharmacodynamics as well. There is network pharmacology. There are a lot of studies for their level of empowerment.
so is there any possibility to collaborate BIBAO if we want in the near future
Yeah,
by
yeah, sure,
connecting?
sure, sure. Sure, I'll be open. I will be happily open to collaborate.
Where we can get your email IDs and mobile numbers, sir, for connect in the near future.
Yeah, yeah, yeah.
Uh,
I can provide the email ID itself. I'm writing it in the chat box. Connect.
I will definitely we will, be in connect in near future. But I used to visit Lucknow, sir, so I
Yeah,
will
sure,
definitely
sure,
want
sure.
to.
Yeah, sure, sure, sure.
Sure. Thank you.
So,
Hello. Yeah,
hello, sir. There is a question raised in a chat box.
yeah.
What is the difference between prebiotic and probiotics? How should we incorporate in daily life, daily diet?
Yeah, 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 yeah. That is a quite a nice question. because as I told you that probiotics are live biotherapeutics, live bacteria that may confer positive effects inside, and the most common bacteria is known as the lactic acid bacteria, which is found in curd. And for probiotic bacteria, other probiotic strains also, we need fermented foods. So fermented foods like idli, kombucha, sauerkraut, and this yogurt, not even curd, yogurt. Use these kind of fermented foods in your diet. And for prebiotics, those are basically oligosaccharides, mono-oligosaccharides, fructo-oligosaccharides. So these kind of fibers you need to include. Garlic in your diet, onions, pears. These should be included in your diet as a prebiotic. You may have popcorn in your diet as a fiber. You may search, there will be a lot of fiber rich diet that may be prescribed. But as I already said, that balance should be maintained, because if you will have lots of fibers and lots of this probiotic, it may cause some kind of gastritis or bloating also. So vitamin B12 is also needed to digest these kind of things. So you need to have a multivitamin also to maintain the balance with pre and probiotics.
Thank you, sir. And also another question. What kind of foodstuff may be responsible for causing cancer?
The major food stuff responsible for causing cancer is the smoked diet, the microwave oven where we use radiations to cook the food, these radiations, and the diets which are having nitrosamines, these these fast food which are being cooked with preservatives. These kind of foodstuffs are responsible for basic have been linked with causing cancer, and still much and much more research is going on, because the preservatives, the and sometimes we use packaged stuff. So the packaging is having some nano particles, the nanoplastics, or the packets that are being made of nanoplastics. Those also gets inside the food, and that goes inside our system. So that is very important that we use fresh food, immediately cooked food. That is much more for us, rather than packaged and fast food and grilled, oven, smoked. These kind of foods should be avoided. And also we use oils that are being reused. Sometimes that become trans fat. Those are trans fatty fat oils. So that is why we say that use the cooking oil once. Don't use it twice or thrice, because that causes from cis fat to trans fat. That may be also linked with oncogenesis.
Thank you, sir. And also another question. Is there any drug food interaction that causes cancer?
Yes. There may be a possibility. That is a very nice question. There are lots of interaction. That is why we are saying that we need to study much more, because food interaction. There are like if I've studied about mushrooms, those are functional food. Those are these macrofungus also. So they are live, and when we pluck it, they become postbiotics. And when we have those fungi, mushrooms are having mycotoxins. So they may confer good effects also as a prebiotic, as a postbiotic also, but at times they may have interaction with the drugs also. So we will be experiencing allergies as well. So there the allergy profile is also need to be discussed. So there are a lot of possibilities. That's two drugs, and drug interaction is much very prominent possibility, and drug food interaction is also a prominent. That is why it is being recommended by a dietitian about which diet you are need to have with a particular medication is going on. And nowadays there is a prominent role of a dietitian for the case of different diseases. When you have cancer, there is a diet plan for you in a cancer regimen, for a healthy regimen. So that is a very important point, and that needs to be. And there is a study or a department in CFTRI Mysore that is working with precision nutrition, or in NIN Hyderabad. So they are also talking about this nutrition and diet in respect to individualized or a personalized therapy. That each individual should have a different diet according to the genomic composition, or according to the prognosis of which disease he may likely to get. So that is a very emerging area of research and emerging area of study.
Yes, sir. And also one more question. During gestation, does the fetus get affected?
Yes, 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 yes. Gestational diabetes, gestational this kind of drug transference. At many studies, there is a transfer of because the immune system and the microbiome transfer is up to the level of feces. That is why it is being preferred not to have C-section or a cesarean section. If we don't have a cesarean section and there is a normal delivery, the microbiome of the fetus will be restored and will have a better composition as compared to having a C-section. So that is why it is being preferred. But nowadays, due to many kind of problems, we used to have C-section. But the best way to have the best microbiome and to have the best immunity, we need to have the normal way of delivery. So that is very, very much effective. And the transfer of the microbiome from the mother to the fetus is through in the womb itself. So for that development, it is very much needed.
Thank you, sir. And one more question. Are nanopharmaceuticals better than currently available options for cancer treatment?
Uh, It may be, it may not be. Because nanopharmaceuticals are very good, they may be targeted to the point, they may confer beneficial effects, but at times they may also cause toxicity. Their toxicity, because nanotoxicity is also posing a lot of threat. So that is why I told you engineering a bacteria or a biohybrid or a biomimicking this natural process is much more beneficial than having a nano artificial means synthetic nanoparticle, because this bacteria, if it is having the cargo, it is we design that bacteria to deliver good drugs in spite of having toxins, secreting toxins. That can be a newer way of delivering this drugs into the target site. So that will be a bio-based method in spite of the synthetic nanoparticles we synthesize.
And also one more question, sir. What is the impact of probiotic available in the market such as Yakult on the health, and it is recommended for daily consumption?
Yes, Yakult is a good thing. There is a lot of more probiotics in culture. Like there are, look, one more thing. Everyone knows that lactobacillus is a good probiotic. Lactobacillus confirms good. You need to have yogurt. It is about bacteria. But lactic acidosis is one of the most important acid found in cancers. So lactic acidosis and the production of more lactic acid is a common implication in cancer. That is why it has been replaced by a fungal probiotic, that is Saccharomyces boulardii. So and also there have been a mixed number of bifidobacterium and other strains also. So it is all about which strain that needs to be given to a particular individual. So strain specific probiotic and is also much being developed in the recent past. So we are working, and everyone else in the country and abroad is working on strain specific, which probiotic is being given to a particular individual. So that precision therapy is also under research.
Thank you, sir. Okay, thank you everyone for active participation. Now I would like to call upon Manoj to present the vote of thanks.
Hello, sir. Can you hear me?
So first, I would like to thank you for delivering such a wonderful session on this topic. Hello.
Manoj, your voice is audible.
So first, I would like to thank you for delivering such a wonderful session on this topic. It is my honor and privilege to conclude this session by extending a vote of thanks. First of all, we are very much thankful to the management, Honorable Vice Chancellor and Pro Vice Chancellor for their continuous encouragement and support in conducting webinars. On behalf of all faculty members of Department of Pharmacology, I thank our Dean, Dr. S. Bharat, for his proactiveness and valuable guidance. And we are really fortunate to have an eminent speaker for today's webinar, Dr. Ishant Pandey. In spite of his busy schedule, he agreed to deliver his talk, and we are very much thankful to you, sir.
Thank you.
On behalf of the Department of Pharmacology, Faculty of Pharmacy, and all the participants, I profusely thank Dr. Ishant Pandey for delivering his talk. I would also like to convey my sincere appreciation to our department faculty members for their support and cooperation throughout the process and the arrangement to successful conduct this webinar. On my personal behalf, on behalf of Department of Pharmacology, Faculty of quickly thank all the participants for their active participation, support, and presence in making this program a noteworthy successful. And thank you, one and all, sir.
Thank you, Manoj.
Thank you, everyone.
Thank you so much, sir.
Thank you. Thank you.
Sir, can we share this complete webinar on our YouTube channel?
Yes. Sure. And please provide me also.
Okay, sir. Thank you so much.